Welcome back to Chapter 5. In this section, we're going to be talking about cloning and genetic engineering. So, cloning, entire organisms can actually be cloned. It's relatively simple if you're dealing with single-celled organisms, but you can actually clone larger species, such as a sheep was actually the first major animal to be cloned. There's actually kind of a niche market for this with, super rich people. They have a pet that dies and they will clone the pet so that they can keep the pet for longer. There are several issues with that as, the organism, the pet is not healthy or as healthy as the original. Its DNA is older, so there are some issues there. And then you don't always get the same personality. So, a clone is just a genetically identical copy of a gene or of an entire organism. So you can clone genes or just segments of DNA, or you can clone the entire organism itself. So cloning does occur in nature. You see it in bacteria. So basically when bacteria divide, it's called binary fission. And the bacteria don't actually cross their DNA with any other bacterial cell, except in instances when the bacteria is under attack from something. And so when they divide, it's actually a cloning process because they produce exact copies. Some plants can actually clone themselves from roots, and then some simple animals can also clone themselves through budding or regeneration. Now that's usually talking about worms or nematoids or something like that. Okay, so or yeast is actually a simple eukaryote that can do this. Basically, they just produce a little bud that spouts off, and then it will eventually be pinched off from the main body, has the exact same DNA as the parent, and then grows up into a fully functional organism. So mammals can be cloned through a process called nuclear transfer. So what they do with this is they take the nucleus from an egg cell. Okay, so they take an egg cell. It doesn't necessarily have to be from the mammal that they are cloning, but it does need to be from the same species. Okay, and they will just remove the nucleus from that egg cell. And then they take the nucleus of a cell from the animal that they want to clone and implant that into the egg. So now you have a brand new cell. Okay, so the egg cell is, doesn't have a nucleus. So they take the nucleus out of whatever organism, cat, dog, sheep that they want to clone. They take that nucleus and that's it, and then implant it into the egg. So now you have a cell that has the DNA from the animal that they wanted to clone. Okay? So at this point, they will actually implant this new cell into the uterus of an animal of the same species and then just wait for it to grow. And that's how they create a clone. So it does have some potential benefits. If you're talking about cloning on a smaller scale, you can clone organs for transplant into humans, because the organ transplant side of medicine is actually really kind of struggling a lot. It always has been, what with the fact that you need to make a really good match with the organ and the person that's getting the organ, that the organs have to come from other humans. So if a person needs a heart or some vital organ like that, somebody else has to die in order for them to get those organs. And then there's always the risk of rejection, organ rejection. So the body will recognize the organ as, hey, that's not part of me, that's not, originally me, and so it will start to try to destroy the organ. So there are a lot of issues with organ transplant as it is today, and so they are working on being able to clone and grow organs from a patient's own DNA in order to help out with that. because that avoids a lot of the issues. Cloning could also, or we're looking into it for saving endangered species. I think the white rhino recently went extinct. So something like that, you could clone the rhino in order to try to keep it alive. Once it gets down to the extinction point, it's kind of difficult to do that because, I mean, you only have the one, and yeah, that's a problem anyway. But cloning does raise some concerns, such as the low success rate, okay? Because the egg cell can kind of reject the nucleus. The uterus that they try to implant it into can also reject it, so there's a lot of kind of iffiness there. The clones are imperfect, so they are not perfect clones. They are less healthy than the original animal. Generally, the DNA is older, and so it doesn't hold up as well. So you will see degradation throughout that animal's lifetime. And you also get a decrease in biodiversity. So biodiversity is important to help keep the health of a species and a population of that species. All right, so talking about genetic engineering. So new genes can actually be added into an organism's DNA. Generally, when we talk about this, we're talking about like bacteria, not humans. Okay, that gets into a whole huge ethical thing. Same with cloning humans, that gets to a whole new level of ethical concerns there. But so when we're talking about DNA from a bacteria, it's usually found in the form of what's known as a plasmid. It's just a single piece of DNA that's connected into a circle. And so through genetic engineering, you can actually insert a gene from something else, some other organism. And it doesn't have to be just another bacteria. For example, in the research that I did in undergrad, we actually added in the genes that coded for spider silk proteins. So we were able to study the proteins on their own without having to deal with the spiders, which, you know, avoided the whole cannibalism thing that spiders have. So yeah. Great. But genetic engineering involves changing an organism's DNA in order to give it new traits. So the example with the spider silk proteins, the new trait we were giving it was that that bacterial cell was able to produce spider silk. Now, sometimes, I mean, you can get a whole bunch of different traits through all of this. And genetic engineering is based on what's known as recombinant DNA. So basically combining two pieces of DNA from different hosts. They do this, they are working on this a lot with plants as well. Haven't really gone into animals, but plants and bacteria are the big ones. So recombinant DNA just contains genes from more than one organism. Okay, so this is cross-species. Again, we are doing this a lot with plants, especially trees, to try to resist the effects of droughts or different parasites that go after those specific trees. This is also how we got human insulin to be produced from bacteria. So we inserted the gene for human insulin into the bacterial plasmid, and then the bacteria species started to produce human insulin, which of course is very important for anyone with diabetes. So bacterial plasmids are often used to make recombinant DNA. It's the simpler form of this technique, simply because the bacterial plasmid is much smaller, and generally we know the entire gene sequence of that plasmid. So plasmids are loops of DNA in the bacteria. They are kind of outside of the main chromosome. So it's kind of a secondary circular plasmid, little loop of DNA. Again, they are much smaller than the bacterial chromosome, okay, which you can see here. the plasmids generally carry genes for things like antibiotic resistance or different things like that the bacteria will oftentimes just kind of, they'll make copies of it and then just shoot those copies out into their environment. And that way other bacteria can come up and say, oh, this might be useful, and just kind of pick it up and start using it and see if it helps. So the fact that you have antibiotic resistance found on these plasmids can cause kind of issues when you're talking about health. If your bacteria have these plasmids that give them antibiotic resistance, it can be difficult to fight that disease. So in order to use these for genetic engineering, first thing they will do is they will actually cut the plasmid with a restriction enzyme. And then they'll also cut the foreign DNA with that same enzyme. So generally when they do the foreign DNA, they've actually sent that to a lab that sequences it exactly the way they want it. So you've got the restriction enzyme sites on either side of the gene that you want to put into the plasmid, and then you have that gene in the middle. Okay? And you also have some kind of way of testing if the gene was inserted into the plasmid. So oftentimes that's also a gene for antibiotic resistance of some kind, or it'll change color of the bacterial colony or something like that. Just to say, hey, we know this plasmid picked up the foreign DNA, and then this bacterial cell picked up that plasmid as well. So the foreign gene is inserted into the plasmid, and then they get fused together. So it's important to note that they are being digested with the same restriction enzyme so that they have matching ends, okay, that can be easily fused together. So genetic engineering produces organisms with new traits. These are generally called transgenic organisms. You might have heard of genetically modified organisms or GMOs. Okay, this is in the same realm here. It is an organism with one or more genes from another organism that's inserted into its genome. Okay, so this is kind of looking at the whole process again, cutting both the plasmid and the foreign DNA with a restriction enzyme, and then fusing it together. So transgenic bacteria can be used to produce human proteins. So I talked about insulin. Okay, so that's an example here. The gene is inserted into the bacterial plasmid. Plasmid is inserted into bacteria, which is actually really simple. All you have to do is kind of shock the bacteria in some way. Usually you do it with heat, and then they just start picking up anything in the environment to try to protect themselves. Bacteria will then express that gene, and the protein can then be purified. Oftentimes that results in having to break open the bacterial cells to get the protein out, but sometimes you can actually modify the gene in order to get the bacteria to kind of push it out of the cells on their own. So insulin is a good example of this, as is human growth hormone, so HGH. It makes the news every once in a while with steroid use, but it can also be used to help people who are behind the growth curve in height. I knew a guy in middle school who was actually, he told everybody he was on HGH because he was under 4 feet tall, I think. I don't remember, but he's normal height now. Okay, so we're actually going to stop here and then pick up with the last couple slides on the next video.